After his presentation, we will open up the session to questions and answers. So today's topic, as I'm sure you're aware, is the Selinexor combinations in clinical trials. And Selinexor was first tested for penta-refractory myeloma patients in 2018, or in other words, patients who had been through multiple lines of previous therapy. Over time, and as a result of successful clinical trials, we have seen XPOVIO's most recent approval in December of 2020 for the treatment of myeloma patients with at least one prior therapy. Selinexor continues to be a focus of clinical testing as we learn more about the drug, what dose is the most effective, how can we manage the side effects, and how frequent it should be administered, as well as what combinations of myeloma medicines along with XPOVIO or Selinexor are proving to be most successful. Today, we're going to have the privilege of hearing from Dr. Zonder as he presents to us what we are learning about Selinexor within clinical trials and what we've learned thus far. Dr. Zonder is a professor in the departments of oncology at the Barbara Ann Karmanos Cancer Institute and Wayne State University School of Medicine. He is the leader of the KCI Multiple Myeloma and Amyloidosis Multidisciplinary Team and co-leader of the Molecular Therapeutic Program. He is a member of the steering committee at the Multiple Myeloma Research Consortium, a medical advisor for the Amyloidosis Support Group Networks, and a member of the International Myeloma Working Group, the International Myeloma Society, and the International Amyloidosis Society. He's a member of the Southwest Oncology Group's Barlogie Salmon Myeloma Committee. Dr. Zonder has authored or co-authored numerous original research papers, review articles, book chapters, and research abstracts on myeloma and amyloidosis. He completed a fellowship in hematology oncology at Wayne State University and his medical residency at the University of Rochester, New York, and he received his medical degree from Wayne State University School of Medicine. As you can see, he is very qualified to speak to us today, and we're really looking forward to your presentation, Dr. Zonder. Thank you for being here.
Thank you so much for the kind introduction. Can everyone hear me okay? Great. I'm going to share my screen. Not start at the end, we'll start at the beginning. Patient talks are the most, patient-focused talks are the most fun for me to give. They're the ones I actually like giving the most, but they're hard because there's varying needs, and there are rookies. There might be people on this call who've been on Selinexor for less than a week, and there might be people who've been troubleshooting Selinexor for the last 4 to 6 months, and you may all need kind of different things, and it's even harder when I can't see everybody. So I'm going to just say right off the bat, because that it's a pretty small group, and we should keep this informal, meaning people can speak up while I'm talking. I can't see you. I don't know if you have a burning question. I actually don't mind if we break it up with questions as we go, or we can save the questions to the end. I'm really pretty flexible about this. These are my disclosures. None of them are Karyopharm, the maker of Selinexor, but I do consult for multiple different companies that make and market multiple myeloma and amyloidosis therapies. So I'm going to talk first about what Selinexor is, how it works. We'll discuss the approved indications. We actually just got a brief overview of the what it's approved for, and we'll talk about that. Then we'll talk about useful off-label combos and the studies that support that. I'll briefly talk about some other open studies that we are waiting for results on, and then we'll finish up by talking about Selinexor toxicity and troubleshooting the management of that. Excuse me.
I'm sorry.
Thanks. Hi. First, let's. What is Selinexor? So, not a very useful picture for you or for me, as I'm not a biochemist. but it's an oral medication. It's the first in class drug in a family of medications called selective inhibitors of nuclear export, or SINEs. And what it specifically inhibits is something called XPO1. So what does that even mean? So I'm going to sort of walk through the mechanism. This is my inner nerd coming out. There are proteins in every cell, but particularly in cancer cells. There are proteins in the nucleus, or the sort of the brains of the cell, where the DNA is and where genes are, gene expression is regulated. And there are proteins that suppress the growth, well, at least suppress or regulate the growth and survival of these cells. And XPO1 is a protein that picks up these proteins and shuttles them out of the nucleus, and it basically functionally deactivates them because they're no longer in contact with the part of the cell that they regulate. And what selinexor specifically does is it blocks XPO1's function. What does that even mean? So graphically, this is a picture, right? So the red diamonds are the tumor suppressor proteins, and this makes it look like there's one kind of tumor suppressor protein. This is really a whole group of different proteins that regulate cell function in a lot of different ways. And XPO1 is the orange V's, or the orange shapes here. And what they do is they latch onto these tumor suppressor proteins and shuttle them through these nuclear pores into the outer part of the cell, which is called the cytoplasm. And they basically become deactivated out there. And then what happens is the XPO1 actually shuttles back into the nucleus through these pores, and it picks up more cargo and shuttles them back out. And it's this rotating cycle. But what happens is selinexor blocks the binding of these cells. And so it renders XPO1 unable to move these suppressor molecules out. And so that leads to, direct cell killing on tumor cells, because tumor cells overexpress XPO1. And importantly, it increases the cell's sensitivity to other therapies. And this is actually, I think, one of the key things. This is the rationale for all the combination studies that we're going to talk about. And so what this picture here is showing us is a graph of tumor cell growth when they're injected in a mouse model of multiple myeloma. And so what control means is that the black line at the top, it shows you how fast the tumor cells grow in the mice if you just inject them and watch what happens. And then the other lines show what happened when you treat those mice with different agents. And you can see here when you use carfilzomib, which is a proteasome inhibitor, commonly used myeloma drug, you can see that you partially suppress the growth of these tumor cells. You get a similar depression of growth when you treat them with selinexor, but when you use them in combination, you get more than additive suppression of tumor growth. And in fact, in this particular model, there was practically no growth over an extended period of time when you use the combination. And research has been done at multiple centers. Wayne State University, Karmanos Cancer Institute, one of our basic scientists here has done a ton of this preclinical synergy work with myeloma drugs, pancreatic cancer drugs, and lymphoma drugs. And actually, we've conducted not just myeloma combination trials, but we've conducted and reported a lymphoma study of selinexor in combination with a standard agent, based on the fact that it was shown to be synergistic like this with anti-lymphoma therapies in cell lines in a mouse model of lymphoma. So the effects of selinexor are not unique to myeloma. You know, there are some drugs like Daratumumab, which specifically bind a target that is really almost only expressed on plasma cells or myeloma cells. Selinexor's effects are much broader than that because all cells really, to some degree, have pathways or sensitivity to inhibition of XPO1. I mean, it's different cells to different degrees, but XPO1 is something that's present in multiple different kinds of tumor types. So a really kind of an interesting drug in that respect. Now, the studies that led to the approval of selinexor, I'm going to show in just the next couple of slides. The first one is the STORM study, which was a phase 2 study. This was basically just selinexor, 80 milligrams twice weekly, plus dexamethasone, 20 milligrams twice weekly, given in repeating 4-week cycles to over 100 patients with relapsed myeloma until they had progressive disease. And the patients on this study had to be refractory or have disease that was resistant to drugs in the Revlimid Pomalyst family. That's the IMiDs, proteasome inhibitors, which is the Velcade or carfilzomib or ixazomib family, Daratumumab or a related CD38 antibody, steroids such as dexamethasone, and importantly, whatever their last therapy was, they had to be progressing on their last therapy. And the primary endpoint of this study was simply the response rate, the overall response rate. And what we saw in this study was an overall response rate of 26%. Now, it doesn't sound like a lot, but importantly, in patients who were really resistant to prior therapies, and it's in red text here, and it was referenced earlier, penta-refractory patients, that means their myeloma has progressed on Revlimid and Pomalyst, Velcade and carfilzomib, and Daratumumab, or a different CD38 antibody. So 2 drugs in the IMiD family, 2 drugs in the proteasome inhibitors, and a CD38 antibody. That's a pretty resistant group of patients. A 25%, basically the response rate for patients in that group was really the same as the response rate for the overall group, even if they weren't refractory to all of those agents. The duration of response wasn't very long on this particular study. It was a little over 4 months. We'll talk more about that later. That is, obviously, that's probably the most, I think, challenging part of this data. Although a significant number of patients respond, many of them are pretty short responses. There definitely were patients on this study who had responses lasting in the 8 to 12 month range, but it was not the majority of patients. In terms of safety, the common side effects that we saw were anemia, low platelets, nausea, fatigue, low sodium, and low white blood cells, specifically the white blood cells that fight infections, which are neutrophils. That's called neutropenia. Dose modification was necessary for both selinexor and dexamethasone on this trial. At the end of the day, 18% of the patients on the study stopped because of toxicity of the medication. The rest who came off study eventually and came off study because of progressive myeloma. This actually led to the approval of the drug. Subsequently, there was another study, or led to an accelerated approval of the drug, which needed to be validated in a subsequent randomized study. And this is the randomized study. It's called the Boston study. And this is patients who were treated a bit earlier. Patients in this study had only had on average 1 to 3 prior lines of therapy. So although they were often resistant to 1 or 2 or 3 of the drugs that I mentioned before, it was very unusual to be resistant to all 5 of them. This was about a 400 patient study, and there was a randomization between bortezomib, or Velcade is the other name for bortezomib. Bortezomib twice weekly plus dexamethasone 4 times per week. That was the day of and day after bortezomib, versus bortezomib once weekly, dexamethasone twice weekly, and selinexor 100 milligrams weekly. So this was a different schedule, right? So in the last study that I mentioned, the selinexor was 80 milligrams twice weekly. This is a lower dose functionally. It's 100 milligrams once weekly. And then patients on this study were treated to progression. What we saw on this study was progression-free survival that was about 5.5 months longer in the combination arm than in the other arm on the study. Progression-free survival means being free of either disease progression or death from any cause. And so progression-free survival was over a year on this study, median progression-free survival. One can argue or debate about the utility of 5 extra months of progression-free survival. I argue that that's worth something, especially when the, there you go. Especially when the toxicity was actually not more pronounced in the arm that did better. There was a higher rate of certain toxicities like low blood counts, but one of the key toxicities that we worry about with bortezomib-based therapy is peripheral neuropathy. And grade 2 peripheral neuropathy is functionally significant, like the kind of thing that interferes with your ability to do day-to-day things like putting in earrings, buttoning your shirt, zipping your pants. Grade 3 neuropathy is worse than that, where you actually have a hard time doing activities of daily living, where things like cooking, cleaning, self-care start becoming problematic. So even the grade 2 is meaningful, and the rate of grade 2 neuropathy or greater was over 10% lower in the selinexor arm, and it was because adding selinexor permitted the investigators to successfully reduce the doses of bortezomib and dexamethasone so that patients enjoyed having less toxicity from those drugs, even as their progression-free survival was actually statistically better. So this study led to the approval of Velcade, dexamethasone, and selinexor in patients who had 1 to 3 prior lines of therapy. Now, we use selinexor with a lot of different drugs, even ones that it's not approved with. And so the following studies that I'm going to briefly talk about are off-label combinations. And they're mostly being studied in the context of this multi-arm study called the STOMP trial, which is looking at selinexor in combination with pretty much everything you can think about here. So it's all sorts of, every drug that's used for the treatment of relapsed multiple myeloma plus selinexor is being studied. And there's even some arms, like arm number 7, where selinexor is being studied in combination with lenalidomide and dexamethasone as upfront therapy in newly diagnosed multiple myeloma. This is a study which over time has evolved. For instance, arm 10, selinexor plus an antibody called belantamab mafodotin, which is also called Blenrep. That is a very recently approved antibody drug conjugate in myeloma. And the original STOMP study did not have arm 10, but it was modified as this drug became available. There's interest in exploring how selinexor can be used in combination with that drug and with others. So pretty much everything you can think of, and we're clearly not going through every arm on this study. I'm going to sort of focus on these 3 that I've put in red, and I'm going to just really briefly talk about some data from each of these. So the first one is the combination of selinexor, pomalidomide, and dexamethasone. Again, this is one of the arms of the STOMP trial. This has been reported as an abstract at various meetings, including the ASCO meeting, American Society of Clinical Oncology, last summer. 65 patients were enrolled in this arm of the trial, and the response rate in patients who were not already resistant to pomalidomide was 57%. So that is, the response rate to pomalidomide and dex is expected to be 25 to 30%. The response rate to selinexor alone is expected to be, or selinexor and dex is expected to be about 25%. So at the least, it's additive. But I would argue that there's actually evidence of synergy. And the reason I'm saying that is because there was a subset of patients on this study who are already resistant to pomalidomide. So you would expect that hardly any of them would respond to treatment if you just retreated them with pomalidomide. And yet when you treated them with this combination of selinexor and pom, response rate was 44%. So that's actually higher than the response rate that you'd expect with either drug by themselves, even if the patients weren't already resistant to pom. So this tells us that adding selinexor restored some measure of sensitivity to pomalidomide. So there's synergy here. And on this trial, there was no unexpected toxicity, meaning the side effects that we saw were the ones we already knew about for selinexor, pomalidomide, and dexamethasone. Dr. Gasparetto reported on a different arm of the STOMP study. This was selinexor, daratumumab, and dex. This has been reported at a couple of meetings and then published after that. 34 patients with relapsed myeloma. The selinexor in this arm was dosed at 100 milligrams. They looked at different schedules. So some patients got 100 milligrams once weekly. Some patients got 60 milligrams twice weekly, along with standard daratumumab. No patients were treated at the 80 milligram twice weekly dose that Seli was originally approved at as a single agent. And the response rate was 73%. This is actually pretty impressive. This data got a lot of attention. Again, the response rate to selinexor, 25%. The response rate to daratumumab is expected to be about 35%. This is more than additive. So this strongly suggests synergy. And the progression-free survival in this group was over a year, which is, again, it tells us that adding dara to selinexor, or vice versa, however you think about it, significantly prolongs the prolongs the average duration of disease control compared to what you'd get out of either drug alone. There have also been multiple different groups looking at the combination of selinexor, carfilzomib, and dexamethasone. This one is the one I'm reporting on here was actually not part of the STOMP study. This was a phase 1 study that we collaborated with University of Chicago on. Andrei Jakubowiak was the lead author. This was a phase 1 dose escalation study where the doses of selinexor and carfilzomib were both explored at different levels throughout the study. Both drugs were used on twice weekly schedule, and the overall response rate was 48%. The most interesting thing about this study was that there were 13 patients whose myeloma was progressing on carfilzomib right before they enrolled on this study. So the last therapy they had was carfilzomib. They were progressing, their myeloma was progressing, and they went on the study where selinexor was added, and bang, 8 out of 13 responded when we added selinexor. That suggests synergy because 8 out of 13 is higher than the response rate you'd expect from selinexor alone. This was a pretty heavily pretreated group of patients, and again, 33, like a third of the patients on the study were actually known to be resistant to one of the drugs in the combination. So not very surprising that the duration of responses seen on this study was short. The duration of therapy you get is not only, the duration of response that you get is not only related to what the combination is, but when you use it. So the Boston trial, which treated patients after only 1 to 3 prior lines of therapy, had longer responses than this, even though carfilzomib is arguably a more potent proteasome inhibitor than Velcade is. I wrote here this last bullet, plan is a randomized trial like the Boston study. I think that is still on, although when I checked clinicaltrials.gov, there was a study proposing this, carfilzomib dex plus or minus selinexor. And it looks like the plans for that were withdrawn, at least the investigator that was originally going to be doing it. So I don't know what the status of that is, but there had been plans in the works for a Boston-like trial where carfilzomib was the proteasome inhibitor rather than Velcade. Last combo I'm going to talk about is selinexor, venetoclax, and dexamethasone. Venetoclax is a drug that is not approved in myeloma. It's approved for treatment of chronic lymphocytic leukemia and some lymphomas. And it turns out that one subset of patients, the 15 to 20% of patients who have a swap, a translocation between chromosomes 11 and 14, usually identified with FISH testing on their bone marrow biopsy. This is a subset of patients who appear to have a higher likelihood of disease that's sensitive to treatment with venetoclax. We and others, investigators at the University of Miami, reported on this at ASH this year. And we have internal data also suggesting that there's synergy between venetoclax and selinexor, and that the synergy may be sufficient that you could even potentially get utility with venetoclax in the other myeloma patients who don't have this translocation. That has yet to be proved in a clinical study, but both we and the Miami investigators found that exact same thing, was that it may overcome the resistance to venetoclax in patients lacking this translocation. And the Miami investigators in their report, in addition to talking about their laboratory findings, described 2 patients who had disease that wasn't responsive anymore to just venetoclax therapy, who started having a response after selinexor was added. So if you go to clinicaltrials.gov and you type in selinexor and myeloma, 30 different trials come up. Some of them are completed, some of them aren't yet recruiting, but basically virtually any combination of therapies that you can think of, like any backbone that you can think of adding selinexor to is being studied. There's a trial adding it to Daratumumab, lenalidomide, and dexamethasone as part of upfront therapy, being headed by a consortium of hospitals. That's also an arm. A similar regimen is one of the arms of the STOMP study. And by the time the STOMP study is done, it'll have been tested in combination with pretty much everything. I want to finish up by talking about selinexor toxicity. I think anybody who's been on the drug knows that GI toxicity can be significant with this drug. That's particularly true with twice-weekly dosing. Anorexia, nausea, and vomiting, which are basically upper GI toxicities, are common. Diarrhea is also seen. Fatigue is extremely common with this drug, and sometimes it's like wipeout fatigue. Hematologic toxicities are seen. Any of the blood counts listed here can be lowered, and that's that's an overlapping toxicity with many of the agents that it's being tested in combination with. So that's something that has to be watched for. Infections are seen, and a unique or specific side effect related to selinexor that we don't see with many other myeloma drugs is low sodium, which is also called hyponatremia. So just to talk about basic principles of toxicity management, and I figure mostly what we're going to do is probably talk about specific instances of toxicity and troubleshooting that. So I don't want to go too deep into this, but this is sort of my general approach, and this is also mirrored in some reviews on the topic. The first one is to set appropriate expectations. You have to really spend the time, more so with this drug than, I mean, we always do that when we're starting new therapies. It is more important with this drug than it is with just about any other drug because the side effects can be so problematic. Getting the nursing team engaged early with patients who are starting this medication, and having an early check-in between the clinic team and the patient to see how things are going really just after the first dose or two, I think is quite important. Patients can run into trouble within a week of starting if they have severe GI toxicity. Antiemetics or medications to prevent nausea and vomiting are absolutely essential with this drug. So on label Zofran, we use Zofran day of and day after each dose of selinexor. Olanzapine is also extremely useful. It's used for neuropsychiatric conditions also, but the dose that we can use for nausea management is quite low. And the combination of Zofran and olanzapine is extremely effective at preventing nausea. So we often use that. The other thing that's useful for managing nausea and GI symptoms in general is to decrease to weekly dosing early if needed. The fact is patients can respond to weekly dosing even when it's used only with dexamethasone, particularly when it's used in combination with other drugs. And so I don't hesitate. I was even commenting, we were chatting before the meeting started. The honest truth is most patients in my practice, I start on once weekly dosing and escalate to twice weekly if I need it and if they can tolerate it. IV fluids are often necessary. If patients are not eating and drinking, if patients have severe diarrhea, they have to be managed with IV fluids. It's also part of the therapy for low sodium. Normal saline can help bring the sodium up in a safe and gradual way. If the low sodium is severe enough, they may need more extreme measures like taking salt tablets. And in that case, I usually involve nephrology and blood work needs to be checked every few days if patients are started on sodium replacement. The other key thing is monitoring weight and calorie intake. Nutrition consults can sometimes be very helpful. We've actually found that we can sort of hijack diet apps that are available on most people's smartphones to help track calories. So we've actually sort of utilized that tech, and it's been helpful for our patients because you can have them enter what they're eating or not eating, and you can have them log in during their appointment, and you can review what they took in every day. So that's something that that we do in practice and that we're exploring more formally, not in a selinexor trial, but in another setting. So to conclude, selinexor is undoubtedly a useful drug. It's helpful. Patients, we have numerous patients that have benefited from the use of this drug, either in combination with dexamethasone or with other myeloma agents. Its applications are going to extend far beyond just multiple myeloma. It's being studied in myelodysplastic syndrome, myelofibrosis, which are other blood and bone marrow conditions, and also solid tumors. Its totally unique mechanism of action is what makes it useful across multiple tumor types. It's also what makes it useful in combination with standard drugs, and doing combinations permit lower, more tolerable doses, not just of selinexor, but as the Boston study showed us, more tolerable doses of the other drugs as well, taking advantage of the synergy. And then, the other thing, as I pointed out in the last slide, the optimal dose and the optimal schedule is variable. And I suspect if we, if people talked about, what dose were they started on, what was their schedule, how quickly was it adjusted, I think, we'll find that it's all over the map, because I know talking with my colleagues around the country, we all sort of manage selinexor and schedule selinexor and dose selinexor a little differently based on our sort of clinical comfort level. And that is my talk. So I will probably stop sharing my screen.
Okay. That was wonderful. Thank you so much.
Thank you.
And
I hope it was not too blabby and.
no, I thought you explained things in a like simple but effective manner. I learned a lot, so I appreciate it. I do apologize in advance because my 2-year old is now home and you might hear him in the background. But I did want to talk to you a little bit. We have questions coming in as well, which is exciting. And
Do you want to share your screen so we can see everybody?
yeah, here, I'll
I'll stop
stop.
sharing. There we go.
Yeah, yeah.
Oh, yeah.
Um, I have a lot of questions for you. Let's start with one of the patient questions. So Joseph was sharing his experience that he's on Selinexor with Dara and Dex. So he asked this before you shared that slide, but it's encouraging to see how effective that is. And again, how long that progression-free survival is really, really encouraging. It was 12.5 months or something like that, if I remember correctly.
The nice thing about that combination is, in contrast to many of the other combos that I discussed and that are being studied in STOMP, the toxicity is less overlapping with these drugs. Dara does have an effect on blood counts. Dara does increase risk of infections, but it doesn't hit your blood counts the way like Revlimid does, or the way carfilzomib does. It doesn't have the GI toxicity that typical chemotherapy drugs do, and so you really can use full dose Dara, with whatever dose of selinexor you'd probably be using anyway, and so it's one of my favorite selinexor combinations. And it's the other thing about it is that even though most of the patients that I treat with that combo have had Dara before, and many of them have had progressive myeloma on Dara before, because we're using Dara so early now, most of that Dara exposure is like in first or second line therapy. And so if you're using like selinexor as like a fourth line drug or a fifth line drug, it's often been a year or two since they've seen Dara, and so it's not, Dara is not exactly a new drug, but it's been a while since their myeloma has seen it, and so I don't know, the combination fits very well into my sort of practice habits, and it takes advantage of the fact that the drugs are just easy to use together.
Yeah, awesome. Thank you. In kind of relation to that, do you see the XPOVIO side effects decrease in drug combinations? So by itself, for example, versus being in a triplet or being with another drug?
I think the reason why selinexor toxicity is less pronounced in the combinations is because the combinations let you use lower doses of it. I usually use 80. I usually start at 80 once a week when I use it in combination with drugs that have overlapping toxicity, with a very low threshold for lowering it to even 60 once a week. When you talk to colleagues, you talk to like Paul Richardson at Dana-Farber, who's a internationally acclaimed myeloma guru. He talks about dosing patients at 40 to 60 when he's using it in combinations. It's really, you're in part trying to get the direct anti-myeloma effect of the selinexor, but you're also really just trying to sort of tip the balance to take advantage of that synergy and help the other drug work better. And so it's, when you're using a dose that's 40 or 60, let's say, again 60 or 80, that's 40 to 50% of the labeled dose of single agent selinexor, right? So yeah, there's less toxicity.
Yeah, that makes sense. Let's talk a little bit more about that synergy, because I wrote in my notes, selinexor is the synergy drug. I loved learning about how even people that were refractory to POM, for example, were able to see results in the trials. let's talk about what it means to be refractory. How does a doctor decide through labs or through other indications that their patient is becoming refractory? And not that I'm doubting the synergy of selinexor, but is it possible that sometimes doctors too quickly take their patients off of a drug and call it refractory? Do you mind talking about that
Yeah.
process a little bit?
So in the real world, the answer to what you just said is yes, right? Sometimes I think that clinicians, may pull the plug on a regimen too early, especially a regimen where there's significant side effects, right? So sometimes it's easier to just stop a regimen than to troubleshoot, like, difficult to manage nausea, anorexia, and diarrhea, right? So, yeah, I mean, I think sometimes there's more that might have been able to be gained, but in the context of a clinical study, there is actually, like the International Myeloma Working Group has a definition for what refractory is. And, so refractory means your myeloma is progressing on or within 60 days of treatment with a specific regimen. And so if that's what it means. And so and the FDA, like when they're evaluating drugs and they're considering the label of a drug, if the indication that's being looked at is, refractory multiple myeloma, multiple myeloma that's refractory to X, Y, or Z, part of their review is verifying that the patients that were treated actually were refractory to X, Y, or Z, to sort of like certify that finding, sort of validate it. So yeah, no, there's an actual definition. So on a study, when they say refractory, a well-conducted study, you can be sure they were actually refractory.
That's encouraging. Thank you.
Mhm.
So Carol brings up a good point, Carl, excuse me. Is it difficult to get approval for off-label use of selinexor with venetoclax? And I had a follow-up question with that. It's surprising to me, I guess it's a small population that has that 1114 translocation, but it's surprising to me we haven't seen the myeloma approval of venetoclax. Do you see that coming in the future? Or maybe as it's proven to be successful with Expovio, as you are theorizing, maybe we see that approval?
Well, so what happened with venetoclax was there was a study that called the Bellini study, which looked at Velcade and dexamethasone with or without venetoclax. And basically what happened on this study was adding venetoclax markedly improved the response rate, markedly improved disease control, and there was a slight excess of deaths in the arm on the venetoclax arm, despite the fact that the myeloma was better controlled. So the issue ended up being infections. And there was it's a great example of a learning curve with a drug. I mean, selinexor has been on a steep learning curve, and my feeling in the myeloma community, like the myeloma research community, is that selinexor's popularity or sort of like traction has increased over the last year or two. Initially, I think everybody's reaction to the side effect profile was, well, this is not a drug that is easy to use. But the fact of the matter is, it is a better drug than some other approved myeloma drugs. There are some drugs which I'm going to get myself into so much trouble, but there are some drugs which have very little single agent activity. I don't mean the 25% likelihood of a response. I mean like almost 0%, so panobinostat, only works if you use it in combination with other drugs. Elotuzumab only works, more or less, only works if you use it in combination with other drugs. Selinexor and the antibody that I mentioned before, belantamab, I mean, they have toxicity issues, but the fact of the matter is they have single agent activity. And so they're going to be used. And as soon as you understand how to troubleshoot that toxicity, the drug becomes more usable. And I think most of us like to use selinexor in combination with other drugs. I mean, I think that that's the emerging trend in the use of this, because it permits the weekly dosing, it permits the lower dosing, and it just makes it a more manageable drug.
Yeah, thank you for that. I saw in some of the storm trial or
Stomp,
arms.
the
Thank
stomp
you.
trial arms. Yeah.
Yes, thank you. That there were some quadruplets with venetoclax. I
Right.
mean, the other one, selinexor, being tested. That's
Mhm.
interesting. Give me your take on that. And do you see that in the future of selinexor?
I don't know. I mean, that's to me, that I was also fascinated by that. There were also, like, when you go on clinicaltrials.gov, there's a POM-CAR Dara selinexor. There's an ELO, selinexor imid. I mean, there's some interesting, there's quadruplets with selinexor added to it, or there, I mean, well, yeah, I guess if you count Dex as part of it, I mean, there are some 4 and 5 drug selinexor combos being studied right now, and I don't know. I'm interested to see in combinations like that, where you're already not using maximal doses of the drugs in many cases. You're generally, like with carfilzomib and daratumumab, for instance, like, you're not using full dose carfilzomib anyway. It's just not, not easily feasible. And if you add selinexor into the mix, it's going to just, drugs are going to have to be lowered again. And the question is going to be, does the synergy, does the benefits of the synergy outweigh the detriment of having to reduce the doses of multiple drugs in the combo? I
Right.
mean, it just, we'll have to see.
Right. And even the side effects that all of them working together could produce in your body.
Yeah.
I mean, not that we haven't seen quadruplets before, but it's just something to consider when you start doing that. You talked a little bit about dexamethasone. Many of the patients I've talked to, and I'm sure the majority of your patients, have a love-hate relationship with Dex, mostly hate.
I was going to say, I have very few. I mean, asthmatics are the only ones with the love
Right.
relationship with dexamethasone. Everybody else, it's pretty much just hate.
Yeah, it's pretty much just hate. Would you consider excluding Dex or lowering Dex doses when used in combination with these
Yeah, so
selinexor?
the original study looked at that. The response rate to just selinexor alone without Dex is quite low. So I rarely, if a patient can't tolerate any dose of Dex, I'm not super enthused about just selinexor by
Right.
itself. But yeah, when you look at, like, so on our the phase 1 that I mentioned of the carfilzomib, selinexor, Dex, the 3 drugs each had a very, very similar rate of having to lower the doses of the drugs. Like, all of them needed to be adjusted at about the same clip. It was all like 35 to 45% rate of having to reduce doses. So Dex was just as toxic as the other drugs, which that is not what they told me in medical school. They
Yeah.
assured me if a patient was too sick for chemo, remember Dex, and it's no.
Wow. Yeah.
Totally turned out not to be the case. It's Dex is real.
You learn more in the real world.
Yeah.
Exhibit 406. So Carl's wondering, do we have any perspective on using selinexor with multiple myeloma with NRA or KRAS mutation? I'm not sure what that
KRAS or NRAS mutations. Not yet, to my knowledge. I mean, well, yes, there is experience with that, because 50% of myeloma patients have a mutation in one of those 2 molecules. So it's inevitable that a large portion of the patients on the studies that have been done have KRAS or NRAS mutations.
Okay.
I'm not aware of any sort of subgroup breakout where they looked at the presence or absence of those mutations to see if that mattered. I don't know. That maybe somebody did that, but I don't know.
Okay.
One thing that's interesting about that question is that it's there is interest in, like, a precision medicine approach to patients with that pathway altered. If they have NRAS, KRAS, or BRAF mutations. BRAF is far less common. It's about 4%, but it's downstream of those other mutations. And so all of those are part of the same signaling pathway. And there's been work done adding BRAF and MEK inhibitors, which are classes of drugs that were developed for melanoma, where BRAF is routinely mutated, like 60% of melanoma patients, or something like 60%. So some high number of melanoma patients have these mutations. So it's a super useful class of drugs for melanoma. And it's a potentially useful class of drugs, or two classes of drugs, for that subset of patients with BRAF mutation in myeloma, and maybe a lot more useful. Yeah, I mean, so the interest is, I mean, if maybe it would be useful in the 50% of patients that have RAS mutations. So there's work being done there. And if it turns out that MEK or BRAF inhibitors are useful for this subset of patients, then that just raises the possibility of yet another partner for selinexor. We don't know, but you'd certainly want to explore that if it turned out that you had targeted therapy for that subset of patients.
Yeah, definitely. That's interesting. Greg is wondering about neuropathy when on combination of Selinexor, Dex, and Velcade. It continues to get worse, and so his clinic has lowered the dose of Velcade, but the numbers increase. The general question here would be, when do you recommend that, okay, it's time to move on, especially with high-grade neuropathy?
So that's a toughie. Right. So I think I have a conversation that sort of alludes to this 2 or 3 times every clinic. How the decision making around continuing a certain regimen or even starting a certain regimen, right? It depends on how you place value on different parts of the equation, right? It's a balance of likelihood of response, expense, likelihood of toxicity. Those are three completely different sort of parameters, right? Three different aspects of therapy altogether. And if one of them weighs heavily for you, that may drive the decision making, but it may not be the same thing that drives the decision making for another patient.
Yeah.
In the case that you just mentioned, this is maybe decided for us a little bit. This is a patient who can't quite find a sweet spot with the regimen. If you lower the dose of the Velcade enough to stop aggravating the neuropathy, then the myeloma starts responding. If you can't get a response with the highest dose of a regimen that somebody can tolerate, it's not the right regimen anymore. Even if you haven't been on it long enough to know if it could work, if delivered optimally, if you've already concluded you can't deliver it optimally, then may not be feasible. We come up against that, and it's sometimes you just don't know until you try. Probably what I would do if it was early, really, really early, I would probably try to just swap out Velcade for a different drug and continue the selinexor, because if you haven't been on it long enough to know that the myeloma is refractory to a selinexor containing combination, I would just find a different partner that's more tolerated than the Velcade.
Yeah, this person started in November of last year, so they've had some time on it.
Plenty of time to know.
Yeah. Sandra was wondering if other steroids can replace dexamethasone and still be effective within the selinexor and Velcade.
Yeah, prednisone, dexamethasone and prednisone work very similarly. Dex is 5 times more potent milligram for milligram than prednisone. So 20 milligrams of Dex a week is the same as 100 milligrams of prednisone a week in terms of expected side effects. So yeah, they're interchangeable.
Okay. Carl is asking an interesting question about if a patient could come to your clinic to get optimized to selinexor and then take that expertise home. Something to say to Carl, take what you're learning in this session and take it to your oncologist or to your specialist and share with them what you've learned and try to get the best treatment for you. I can't emphasize enough the importance for you to advocate for yourself. So proud of you for coming here, for educating yourself, and now go take that knowledge to your treating physician and show them that you want to have an educated conversation about this. And I think they'll
Right.
be impressed. And if they're not, you need to find a new doctor.
Yeah. I mean, we do that. Sometimes my role, at the tertiary cancer center in town is not to assume management, but to help troubleshoot. And
Okay.
so we always talk about, do you want to start coming here to be managed, or do you want me to just send recommendations back to your doc, or do you want to do something in between? And we do with complicated drugs like Selinexor or Blenrep. We often do that middle route where, patient's going to go back to Dr. X or Dr. Y and get their therapy at the either on label or some modified regimen, but we're going to have a check-in, like 4 weeks in, 3 weeks in, like pretty early on, just to talk about how it's going and make sure we're still on the tracks, and then keep going with your doc. But we provide some input on just troubleshooting toxicity, and
That's great.
that's a helpful role, and it's one I'm really happy to. I think most of us are happy to serve that function for our colleagues in the community.
Good.
Yeah.
Awesome. That's great. Um, and then Carl's last question is neuropathy temporary with Velcade, or is it or permanent, like with platinum-based treatments?
It depends on the patient. It also depends on how you respond to the neuropathy, meaning, so they looked at this actually. So there was, there was a clinical trial, the VISTA study, and on that study, which was melphalan, prednisone, and Velcade, if you followed the protocol, they actually looked at like what were the outcomes on neuropathy if you followed the protocol guidelines on dose adjustment of the Velcade versus if you didn't and just did whatever you wanted, which was permitted, but not, again, it wasn't the recommended course. And it turns out there was a difference, like, and what the difference was that on average, the patients who followed the guidelines reduced the doses more. So the key with Velcade neuropathy is, cutting back from twice weekly to once weekly, or cutting down the dose from 1.3 milligrams per meter squared to 1 or 0.7, or take a break until neuropathy gets better. Switch from IV to sub-Q if for whatever reason you're still getting IV, Velcade. All of those measures make neuropathy less likely to progress. If you just sort of the published experience is that if you just take a break when neuropathy reaches an intolerable point, in about a third of patients, it just stays where you left it. In about a third of patients, it gets better but doesn't resolve. And in another third of patients, it actually resolves over time. But it can take months. I mean, sometimes it takes months, sometimes it takes a year for neuropathy to really completely resolve. So, my willingness to push on in the face of neuropathy with Velcade, I think, like for many of us, is it depends on a patient's understanding of the risks and also the available alternatives. I mean, sometimes you're over a barrel, and the medications that you have as your best options have certain toxicities, and you just have to slog through them. But most of the time, that's not the case, and you just have to be aware of toxicities. That's so key. I mean, with Selinexor, like really understanding the toxicities and understanding how to not just respond to, but to head off before they even begin the GI stuff. That's the one that you really run into trouble. If a person, if by the middle of week 1, they can't eat, they can't drink, they can't control their diarrhea, they can't control their vomiting, those are the patients that end up dehydrated, hyponatremic, and bedridden. And we saw that early on with this drug before we really knew how to manage it. I mean, it was rough. The beginning was rough, but you know, we got the hang of it with repeat, with ongoing work. And
Yeah, and we continue to learn, right? As
mhm,
more of these
mhm.
trials are giving us results. One of the other things I wanted to talk about before we finished is that one of the toxicities that isn't physical, well, could be physical, but is financial toxicity. Karyopharm has a payment program that is open for patients that are taking selinexor. We know it can be a costly drug. So in our resource email that we send out to all registrants, we'll include the information for that payment program so that you don't have to be covering the cost of selinexor. You can look into your options and see what works for you.
I
And
think
they're more
it's
than willing to
it's a
help.
copay coverage program, isn't it, for the drug?
Yeah, it's called
Yeah,
Carry Forward. And
yeah,
I don't remember a ton about it, but we had a session about it that I'll include in the follow-up email, and I'll include the website as well, because we don't want you to experience financial toxicity either.
yeah. And that's such a thing with all the oral drugs. The degree of coverage for oral drugs tends to be less than the degree of coverage for IV and sub-Q drugs. And so Velcade or selinexor is just one more drug on the list of things
Yeah,
with Pomalyst and lenalidomide, and Exasamib, just one more drug that it really can lead to financial toxicity for patients.
yeah,
And it's that's so real. Some of the combos that I sort of just didn't really talk about. I mean, like, imagine the potential financial toxicity of a, Pom, Ixa, Dex, Selinexor. I mean,
mm-hmm.
I mean, that could be
Impossible.
thousands of dollars of copay, per
Yeah,
month.
yeah, yeah.
So yeah,
Yeah, well, thank you so much, Dr. Zonder. You're getting praise in the chat of how well you did, and I
thank
echo
you.
those words. Thank you so much for the time that you took.
Thanks so
We're
much.
Yeah, we're gonna end with just a couple outro announcements, and then we can finish up for today. Our next meeting will occur in April, excuse me. We're going to be continuing this conversation about selinexor and backbone treatments for myeloma. We're going to be showing you how you can register for some of these clinical trials that are open. And we're going to be explaining a little bit more in detail some of the 11 arms that are open. And then if you're interested in other myeloma crowd community events, on the 16th, tomorrow, is Know Your Myeloma Labs. So if you're getting your labs or discussing them with your doctor and you're still not sure, you can learn more about what you need to know and what's important numbers to follow tomorrow at 1 p.m. Mountain. That's Mountain because it's a geographic chapter for the Mountain West area, but you're welcome to join from anywhere in the world. That meeting will not be recorded, so make sure to attend live if you want that information. The 17th, we're going to hear from Tony Newbern as he shares his stem cell transplant experience with the newly diagnosed myeloma patients chapter. And on the 23rd, at 7 p.m. Eastern, we're going to hear from Dr. Terry Golombek, and she's going to be speaking about the role of supplements in blood cancer patients' health. The link to sign up for any of those events and even more events I haven't mentioned today is found at the bottom of the slide and will be sent out in the follow-up email within 48 hours of the event's conclusion. Another thank you to our sponsors, Bristol Myers Squibb, Amgen Oncology, Genentech, Adaptive Biotechnologies, Sanofi, Janssen Oncology, Takeda Oncology, and AbbVie. And we thank you for helping us build a strong myeloma crowd community. We appreciate you. And thank you again, Dr. Zonder, and hope that you have a great rest of your night.
Thanks for
Thank
the
you so
invitation to
much.
speak. I really enjoyed it.
Yeah, bye-bye.
Bye.